Okay. Maybe let's get started. Hey everyone, thank you for joining us for the Statistical and AI Methods for Health Data Science seminars. This is organized by the StatSub AI group. The group aims to organize activities that empower statisticians to participate more actively in AI research and leadership. Now the group is officially recognized as an ASA interest group. Here is the website, and then we will also upload the seminar recordings to this YouTube channel. And then this is the this is the steering committee and then also the inaugural officers. Actually, the current inaugural officers are finishing their term, and then I want to just give them a special thank. And then without their efforts, this will not be happening. And then it is my great pleasure to introduce today's speaker, Dr. Tingting Zhang. Dr. Tingting Zhang is a professor of statistics at the University of Pittsburgh. Dr. Zhang's research focuses on developing efficient, scalable, and robust statistical methodologies for the analysis of large-scale multimodal brain imaging data. Her work lies at the intersection of statistics, neuroscience, and data science with the goal of advancing our understanding of brain organization in healthy population and then the mechanisms underlying neurological disorders, particularly Alzheimer's disease. Her methodological contributions, including statistical modeling and inference for high-dimensional neuroimaging data, integrative analysis of multimodal data sets, and then techniques for extracting meaningful patterns from complex brain networks. Dr. Zhang has published extensively in leading journals in statistics and neuroimaging. She serves on the editorial boards of premier journals, including as an associate editor for the Journal of American Statistical Association and then the Annals of Applied Statistics. So before I turn it to Tingting, just some logistics of the seminar. So please save your question to the end of the webinar or type it in chat, and then I will monitor the questions. And then we should have some time at the end of the webinar for Q&A. And then Tingting, it's all yours. Please take it over.
Okay, sure. Thank you. Can you see the screen?
Yes.
Okay, great. Okay, first, I would like to express my sincere appreciation to Yi, Edgar, and also Pan for their great devotions in organizing this webinar series. I attended almost all the past talks and have learned a lot from past speakers. So that's why I feel very much honored to share my recent research here. And of course, most importantly, thank everyone for attending my talk. So today I'm going to talk about the brain map correspondence. So this topic has not been extensively studied in the field of statistics, but it has been extensively studied inside the neuroimaging or neurology. So let me first explain the background. So due to the rapid advancements and the wide use of neuroimaging and large-scale data acquisition technologies, we are able to generate enormous amounts of different whole brain measurements of different brain features. In neuroimaging field, we call this whole brain measurements of a certain brain feature as brain maps. So we can have whole brain measurement of structure features. So we have structural brain maps, and we can also have functional brain maps. With PET, we can also produce brain maps of glucose metabolism and also very famous brain measurements of amyloid beta and tau used in the Alzheimer's disease diagnosis. So because of the time, so in this talk, we mainly use the simplest structure and functional brain maps to explain the existing studies and the existing methods and to motivate this project. So let me start with an example of structural brain maps. A lot of neuroimaging studies have been focused on cerebral cortex. So it is outmost a large layer of gray matter. So it's highly folded. Using structural MRI, we are able to measure the cortical thickness at every brain locations, and then we can produce this cortical thickness brain maps. So why cortical thickness is important? This is because it is a very important structural feature that changes over our lifespan. So for example, for babies aged between 0 and 2, their cortical thickness rapidly increases. And then start for children to adolescents with the cortical thinning process. Then for the Alzheimer patients, so some studies show that in the early stage of Alzheimer diseases, we observe cortical thickness first increases. Then at a later stage, advanced stage of Alzheimer diseases, then doctors observe cortical thinning process. So clearly, cortical thickness is a very important structural features of the brain. And based on it, we want to know how it's related or determine normal, abnormal function of the brain. Of course, we can also measure other structural features. For example, we can have this measurement of soccer depth. So we have this soccer depth brain maps, or we can also measure surface area, curvature, gyrification, and many others. With the structural brain maps, of course, we can also have functional brain maps. Typically, functional brain maps are generated by analyzing task-based fMRI data. So typically, in the task-based fMRI scan experiment, so a human subject is put inside the fMRI scanner. So during the scanning, so the subject is asked to perform certain tasks. So for example, in the language task fMRI experiment, so the subject is asked to think about a word. So then this language-based task actually motivates the subject's brain activity changes over time. Then by analyzing this language task-based fMRI data, we can generate language task-based brain maps. So for these brain maps at every brain location, we have a measurement of the extent of brain response to the language task. So by analyzing language task brain maps, we can identify brain regions that are either associated or involved in the language function of the brain. And of course, we can design many different types of task-based fMRI experiments. The most common one is actually motor task. And this is the most or oldest, I think, a task-based fMRI experiment. So in this fMRI scanning, so the subject is asked to perform finger tapping. So by analyzing this motor task-based fMRI data, we can create this motor task-based brain maps. And so the measurement at every brain location it reflects the extent of brain activity in response to the motor task. And of course, there are also many other functional brain maps obtained from different types of task-based fMRI experiments like emotion and memory and many others. So in addition to these structural brain maps and the functional brain maps obtained through using magnetic resonance imaging, we can also get other brain maps using different imaging technologies such as PET. So we have this kind of glucose metabolism brain maps using the PET, and we can also have brain imagings of the brain maps of amyloid beta and tau. These are widely used in the Alzheimer's diseases diagnosis and treatment. And of course, there are also many other imaging technologies that can be used to generate other different types of brain maps that measure different brain features. You can see that, so given the modern neuroimaging technologies, we have enormous, diverse, and complex brain maps that measure different features of the brain. And then a natural question would be, so what's the association between those brain features? What is the association between these brain maps? So these are very natural and also very important questions. In the field of the neuroimaging, we call this studies of brain map correspondence. It's actually just study of association relationship. The method of the brain map correspondence is very intuitive. So typically, people just use the Pearson correlation between brain map measurements across different brain locations as an estimate of brain map correspondence. So for example, if you have a cortical thickness brain map and a sulfur depth brain map, you just calculate the Pearson correlation of the cortical thickness and sulfur depth measurements across all different brain locations as an estimate of the correspondence between cortical thickness and sulfur depth brain maps. So that's very straightforward. The problem is how to evaluate the statistical significance of this Pearson correlation value. Initially, scientists just ignore the spatial correlation between the measurements across different brain locations. And under these independent assumptions, it is very easy to derive the null distribution of this Pearson correlation estimate. So that under the null hypothesis that there's no correspondence between the two types of brain maps. And quickly, scientists find that brain maps have a strong spatial autocorrelation. If we ignore this spatial autocorrelation, so typically this procedure gives us a highly inflated type 1 error. So now the question is, how can we have a more appropriate evaluation of the statistical significance of the estimate of brain map correspondence while accounting for the spatial autocorrelation within these brain maps? So it turns out that after quite a few years, so this is very interesting, all the popular methods are non-parametric permutation-based methods. So this includes spin test, ring smash, and spice test. And each of them has been cited hundreds of times, and overall they have been used in thousands of studies. And I will explain each of these tests one by one. You will see that these tests use different test statistics and also use different permutation procedures to generate the null distribution of the test statistic under the null hypothesis that there's no brain map correspondence. So despite their differences, the intuition are highly similar. So the idea is that they want to use a permutation procedure that can preserve the spatial autocorrelation within each type of brain map while disrupting the spatial alignment between different brain maps. Then these permutation procedures can generate the null distribution under the null hypothesis that there's no brain map correspondence. So let's start with the spin test first. So you will see what's the meaning of that. So here we use a notation, and also under a very simple settings. So assume that each subject has only two types of brain maps, X and Y. You can think X map is a cortical thickness brain map, Y is the sulfur depth brain map. And if we take average of the brain map values at every brain location across all subjects, then we get this group average brain maps. And so the group average is taken at every brain location across all subjects measurements at the same location. Then so for the spin test, they actually use a Pearson correlation between group average brain maps as the measure of the brain map correspondence. So here this correlation, the subscript V indicates that this correlation is calculated across different brain locations. Then to evaluate, so to evaluate the statistical significance of this correlation between group average brain maps, the spin test just basically rotate the brains. So the idea is taking one brain maps out, the group average Y map out. So it first inflates this group average Y map onto a sphere. Then they apply a random rotation to this sphere and project back into brain space. So we have this rotated group average Y map. By this random rotation, So the idea is that by the random rotation, so the overall autocorrelation within the group average Y map is still preserved. But after rotation, the spatial alignment between X map and Y map is disrupted. So if we calculate the correlation between this group average X map and the rotated group average Y map, this correlation should be considered follow the null distribution of no correspondence between X and Y map. And the spin test actually performed this rotation many times. So each times after each random rotation, so each time after each random rotation, you get one randomly rotated group average Y map, and you calculate this randomly rotated group average Y map correlation with the group average X map. Then, so after many rotations, like 1,000 random rotations, you get a bunch of group average, the correlations between rotated group average brain maps. So because you think this correlation obtained through group averaged and rotated group average brain maps are considered follow null distribution of no correspondence between brain maps. So we can use these correlations to evaluate the null distribution. So the histogram, so here is a histogram of this correlation obtained after rotations. Then we can compare the observed correlation against this distribution and obtain a p-value. So if we find that the observed correlation is very different from the distribution of the correlations obtained through rotation, we would reject the null hypothesis that there's no correspondence between X and Y. So this is how spin test is performed. Brain smash also, so brain smash is actually based on another permutation procedures. So brain smash also, first it also uses a Pearson correlation between group averaged X and Y map as a measure of the brain map correspondence between X and Y maps, but it uses different permutation procedures. So it first take out one group average brain map, say group average Y map. So it randomly shuffle the values of the group average Y maps on different brain locations. And then next, it applies spatial smoothing to those randomly shuffled values at different brain locations. And they also add some IID Gaussian noise to each locations. Then they got this brain smash gets one surrogate map. The extent of spatial smoothing and also the amount of noise are carefully chosen so that the overall autocorrelation within the surrogate map is exactly the same of the group average Y maps. Then you can think that, so this surrogate map still preserves the overall autocorrelation function within the group average Y map, but its spatial alignment with X brain maps is already disrupted. So we calculate the correlation between the surrogate map and the group average X map under the belief that the spatial alignment between the surrogate map and the group average X map is disrupted. Of course, you would think this correlation follows the null distribution of no correspondence between X and Y map. And the brain smash perform this random shuffling and the spatial smoothing independently many times and generate many independent surrogate maps. And we can calculate the correlation between this group average X map with this surrogate map and get a bunch of this correlation based on the surrogate map. And of course, as mentioned that this correlation based on the surrogate map are considered to follow the null distribution of no correspondence between X and Y maps. So we can use its histogram to obtain the null distributions. Then we compare the observed correlation between the group average X and Y map against this null distribution and calculate the p-value. So if the observed correlation is very extreme compared to this null distribution, we would reject the null hypothesis that there's no correspondence between X and Y brain maps. So this is how spring smash is performed. Brain smash was developed later than the spin test. Then the more recent permutation-based method is spice test. And spice test use totally different test statistics and also totally different permutation procedures to evaluate the significance of brain map correspondence. So the idea here, so for the spin test and the brain smash, it calculates the Pearson correlation between group average brain maps at the test statistics. But here for the spice test, it actually calculates the average correlation between individual subjects, X and Y brain maps. So here, for each subject, we have X and Y maps, so we can calculate the correlation between each individual subject's X and Y maps. Then we take an average across those subjects' correlations. So this A0 is our test statistics for the spice test. Then to evaluate the significance of this test statistics, the spice test also uses a totally different permutation procedures. So instead of generating surrogate map like the spin test or brain smash did, it actually randomly shuffle the subject label of Y map. So after each random shuffle of the subject label of Y map, then the spice test calculate the average correlations of randomly paired subjects X and Y brain maps. So in other words, the idea is that if I randomly shuffle the subject's label of Y maps, then the spatial autocorrelation within each individual subject's Y map are still preserved, but the spatial alignment between X and Y map are disrupted. Then these average correlations of randomly paired subjects X and Y map are considered to follow the null distribution of no spatial correspondence between X and Y map. So so we perform this random shuffle many times, and each time we calculate the average correlations between randomly paired subjects X and Y maps. And based on this many correlations, and we get the null distribution. So we compare the test statistics against this null distribution and calculate the p-values. So again, if the test statistic is very extreme compared to this null distribution, we reject our null hypothesis of no correspondence between X and Y maps. So overall, you can clearly see that, okay, for these three most popular methods for evaluating brain map correspondence, you can clearly see that they use different test statistics. They also use different permutation procedures to generate the null distribution. Then a natural question would be, are they the same? Do they give us the same results? And of course, the answer is no. We don't get the same result from this method. So here, let's see our real data analysis example. We just use the simplest cortical thickness and circle depth brain maps from the Human Connectome project. So this data have been analyzed for many times. So for this data, you can clearly see that the spin test and brain smash get insignificant results. Those are the p-values. So these are the p-values of the spin test and brain smash. However, spice test gets very significant results, so extremely small p-values. Then the question would be, okay, why they are different? Which method we should use? And how should we associate those different results output by the different methods? So in fact, this is a long-lasting question inside the field of the neuroimaging studies. So researchers are very puzzled by those different results. So despite that, they actually perform a lot of studies or comparisons. So this is a very highly cited review paper. So researchers, the authors actually perform a lot of studies, comparisons of existing method for evaluating statistical significance of brain map correspondence. They actually use a real data like human connectome projects data to compare those different methods. And of course, the conclusion is like one method may tend to be more conservative than the other, and they have different results. The most important conclusion is that we still need statistically rigorous methods for comparing brain maps and also to understand the differences of those different hypothesis testing procedures. It turns out that we need statistical models for brain maps so that under this unified framework, we can rigorously can compare those different permutation methods and understand why they lead to different results and what are the underlying reasons for their association, their relationship. And this is one biggest limitations of those existing methods. A second very crucial limitation of those non-parametric methods, which is also a common issues of non-parametric method, is that if you use this method, you only get a binary answer, yes or no. There is a statistically significant brain map correspondence, or we couldn't find any evidence or supporting the brain map correspondence. However, most of the time, if we reject the null hypothesis, scientists really want to know, okay, to what extent the two different types of brain maps are associated with each other. And we really want to know what are the underlying reasons that drive this brain map correspondence. So at least we need to construct a confidence interval to understand the extent of brain map correspondence. So unfortunately, those non-parametric methods cannot provide those detailed inferences about the brain map correspondence. So to address these crucial limitations in the existing methods, we develop new statistical models for brain maps. So before I introduce our statistical models, let me introduce some notations. We let XVS and YVS denote the observed values at brain location V in two brain maps X and Y of subject S. So S indicates subject, V indicates brain locations. And the total number of brain locations capitalized V depends on the brain parcellation you use. So typically V can vary from several hundred to like 32,000 if you use a vertex level brain maps. And the total number of subjects capitalized as is around 1,000 based on the human connectome project data we use. And if we under this model, so group average brain maps are denoted by X bar V and Y bar V. So this indicates that, so in group average brain maps, so the value at each brain location V is an average of all subjects' brain map values at that location V. And we think that, so using this notation, we believe that the confusion about the existing permutation methods is due to the confusion regarding the different types of variability inside the brain maps. So under this notation, you can see that there are two types of variability in brain maps. One is variability across different locations, that's a spatial variability. The other is the variability across different subjects S. So that's a variability inside the population. So to address this confusion, we basically just use a very classical two-factor mixed effect model to disentangle different types of variability and also to categorize different types of variability inside the brain maps. So here, so this is our models. So here in these models, mu x and mu y are constant. They are we call them global means of brain maps x and y. So they are basically the average of all brain map values across all locations and also across all subjects in the population. And then we also have this alpha xv and alpha yv. So those are the fixed values. Those are basically the location-specific deviations from the global means of x and y brain maps at the location v in the population mean brain maps. You can see, so alternatively, basically mu plus alpha denote the population mean x and y maps. They are the limit of the group average brain maps as the number of subjects goes to infinite due to the law of large numbers. So due to the law of large numbers, so this limit is fixed. So that's why we think that given the fixed brain isolation, the brain locations are fixed. So those location-specific parameters are considered as fixed parameters. They are constant. So here we call this alpha as a main spatial effect because they denote the location-specific deviation from the global means. You can also think of them as parameters to categorize the spatial variability inside these population mean brain maps. So with the population mean brain maps, of course, we have subject-specific features. So first we have this beta xs and beta ys, and you can think of them as a subject-specific whole brain shift from the population mean brain maps. So it denotes the subject-specific brain average features. And because these features are unique to the subjects, and the subjects are considered randomly sampled from the whole population, so that's why we treat all the parameters associated with subject as random because they are randomly sampled from the population. And in addition, we call this beta as a main subject effects because it's a whole brain subject-specific whole brain shift from the population mean brain maps. And we also have interaction effects, so which is parameterized as an interaction between this eta and omega. So eta depends on location only. So it's a fixed parameters. So we call them interaction spatial effects, and omega s x and are associated with the subjects. So they are random variables, and we call them interaction subject effects. So this product categorize subject-specific brain map structural deviation from the population mean brain maps. So after this, we also have the residual terms. The residual terms categorize the variability in subject-specific brain maps that cannot be captured by those main spatial effects, main subject effects, and interaction effects. And to better understand this model, so here I show a visualization of this two-factor mixed effect model. For each individual subject brain maps, either X or Y, so each individual subject brain maps equals the population mean brain maps plus this subject-specific brain average features, or you can also think of this like subject-specific whole brain shift, and also plus this interaction term, which characterize subject-specific brain map structure deviation from this population mean brain maps, and also the residual terms that characterize the variability in subject-specific brain maps that cannot be accounted for this main spatial effect, main subject effect, and interaction effects. And under this model, once we are able to write down this unified model for the individual subject brain maps, we can clearly characterize different types of variabilities. So parameters that are associated with location V characterize the spatial variability in the brain, and the parameter associated with the subjects characterize the variability across different subjects. So with this parameterization, we are able to specify the null hypothesis of different permutation tests. So it turns out that the spin test is targeting correspondence between population mean brain maps, mu plus alpha, but it relies on a very strong mathematical assumptions. So if the population mean brain maps, population mean X and Y brain maps are spatially stationary, and the number of brain locations in the brain maps is large, then the null hypothesis of the spin test is the correlation between population mean X and Y brain maps equals 0. So that's exactly what the null hypothesis we want to test. However, the problem is that, so if the spatial stationarity is violated, or the number of brain location is small, then this spin test cannot reject the null hypothesis for a range of non-zero correlation values. And this range of non-zero correlation values are determined by the correlation between rotated population mean brain maps. Intuitively, you can think if the spatial stationarity does not hold, so the correlation between rotated population mean brain maps will no longer be 0. So that's why the spin test cannot reject the null hypothesis for a range of non-zero values. And since the brains, so next for the brain smash, since for the brain smash use exactly the same test statistic as the spin test, so it pretty much have the similar properties as the spin test. So if the population mean X and Y brain maps are spatially stationary, and the number of brain location V is large, so the null hypothesis of brain smash is that the correlation between the population mean X and Y brain maps equals 0. And again, if spatial stationarity is violated, or the number of brain location is small, so brain smash cannot reject this null hypothesis for basically have 0 power for a range of non-zero correlation values. So after all these mathematical derivations, so we realize that the spin test and also brain smash relies on a very strong and unrealistic assumptions about our brain. Since our brain is a highly structured organ and system, so it can never be spatially stationary. So that's why we think we need to develop a new method that can get around the spatial stationarity assumption. So we think the most straightforward method is bootstrap. All statisticians know the bootstrap method. So we basically perform non-parametric bootstrap procedures to evaluate correspondence between population mean brain maps. The advantage of this non-parametric bootstrap procedure is that it does not make any assumptions about the population mean brain maps. It only assumes that each individual subject's brain maps are independent from each other's. So we perform these bootstrap resamples, and based on each bootstrap resamples, we can calculate this the group average correlations and get a bunch of correlation based on the bootstrap resamples. Based on these correlations, we can construct a 95 confidence interval for the correlation between population mean brain maps. With this confidence interval, we can easily perform the hypothesis testing. So if the 95 confidence interval does not cover 0, we reject our null hypothesis of 0 correlation between population mean brain maps. So now let's see some simulations just to verify our mathematical derivations. So first, we purposely generate the population mean brain maps from spatially stationary Gaussian random fields. And we also set the spatial autocorrelation at a very low value at 0.5. And we also choose a very large the number of brain locations, V equals 32,000. And here we purposely vary the correlation between population mean brain maps from minus 0.2 to positive 0.2. So then based on this, we randomly generate independent data set. For each data set, we apply our bootstrap-based method on brain smash and spin test and calculate the rejection rates of these three different methods versus different correlation values. So the rejection rate at correlation equals 0 correspond to type 1 error, and the rejection rate at non-zero values correspond to the power of this method. So you can see that if the underlying population mean brain maps are spatially stationary, and the number of brain location is large, and the three methods perform pretty well and have very similar performance, we can achieve very high power at a very small correlation value. However, so if for the exactly the same simulation, so if we reduce the number of brain location to 500, and now you can see that the brain smash and the spin test and correspond to the blue and the red curves have 0 powers for a range of non-zero values. And even though our bootstrap-based method can still perform pretty well. So you may ask, how about we always choose a very large number of brain locations so that we can always have a very high power for brain smash and the spin test. So it turns out that the number of brain location needed in order to achieve very high power of brain smash and spin test depends on the extent of the spatial autocorrelation. So for example, here, we actually use exactly the same simulation models, but we increase the spatial autocorrelation to 3. And we still use a very large number of brain location, 32,000. Now you can see that the brain smash and the spin test have 0 power for a range of non-zero values. So this exactly match our mathematical insight. But these are not the most crucial one. The most crucial one is that in real life, our population mean brain maps are not spatially stationary. So here we actually perform a simulation. We generate data that are very similar to our real data. So in these cases, so the population mean brain maps are the same as the group average brain maps based on 1,000 subjects. So in these cases, the population mean brain maps does not satisfy spatial stationarity. And then you can see that even with a very large number of brain location, 32,000, brain smash and spin test still have a 0 power for a range of non-zero values. And so what's a problem for that? So here is the real data analysis result. The real data analysis result is more concerning. So here we just directly use a human connectome project data to evaluate the correspondence between population mean, cortical thickness, and sucker depth brain maps. And you can see here, So you can see here, for the spin test and the brain smash, they get insignificant results when you use a brain population with a small number of brain locations. But if you increase this number of brain location to 32,000, and the spin test and the brain smash have a significant result, even though those correlation is not that dramatically increased. In contrast, the bootstrap-based method always get us significant result and invariant results across different choice of brain populations. And so now here we basically compare the spin test and the brain smash. So you may ask why I didn't compare the spice test. So this is because under our models, we realize the SPICE test is evaluating totally different form of brain map correspondence. So the SPICE test is no longer targeting association between population mean brain maps. It's actually targeting the correspondence between individual subjects' brain maps. So under this model, we are able to derive the null hypothesis of SPICE test. So this is the mathematical representation. It basically say that so the sum of this interaction term and the residual terms in each of brain maps are independent from each other across all pairs of brain locations. And so this is a mathematical representation. To how to understand it? This basically say that the SPICE test is trying to evaluate whether the subject-specific spatial variability in each individual brain maps are independent from each other. So in other words, the SPICE test is evaluating whether the subject-specific spatial variability in two different brain maps are associated with each other or not. But the problem is that you can clearly see that in this null hypothesis, the SPICE test is not evaluating relationship between this beta. And so the beta here represents the subject-specific brain average features. So the SPICE test is only evaluating subject-specific spatial variability across different locations. It's not evaluating the relationship between the subject-specific brain average features. So that's why we think like we also need to evaluate the correlation between the beta, and under this model we need to evaluate different forms of this brain map correspondence. So here under this model, so we basically trying to estimate different types of correlation. So this is the correlation between beta. So that's the correlation between main subject effects, and the correlation between omega, that's the correlation between interaction subject effects. And we also have the correlation between residual terms and also the cross terms. So the last four correlations, so why we have this cross term? This is because if the last four correlations all equal 0, it corresponds to the null hypothesis of the SPICE test. So in addition to the SPICE test, we also evaluate the correlation of the beta. And using the bootstrap-based method, it's very easy to perform the bootstrap-based inferences to estimate this, to evaluate the variability and to estimate these different types of correlation and associated with the different forms of brain map correspondence. And we can construct the confidence interval for these different types of this correlation, and also to perform the hypothesis testing for each of these correlation values. Okay, here let me show you the simulation analysis result. So due to limited time, I mainly tell you what we find. So first one, this one, we simulated the data where the correlation, so the individual subject brain map correspondence purely coming from the correlation between beta, that is the subject-specific mean and average brain features, and all the correlation between residual and interaction and subject effect equal 0. So this plot shows the rejection rate of the SPICE test and all the different bootstrap-based method. So the SPICE test correspond to this blue curve. It's here. It has a 0 power. So this basically confirms that if there's any correlation between subject-specific average brain features, SPICE test couldn't detect that. And however, even though the SPICE test can also have non-zero power for detecting the correlation between the interaction subject effect, that's omega, but the power is very low, indicated by those blue curves. So you can see that. So actually, if the correlation between the individual subject brain maps is driven by those this interaction subject effects, the spice test actually has very low power. So what do we find that? Actually, the spice test is most sensitive to detect the correlation between residuals, and it can achieve 100% power, even though those correlation between residual is very small. So and this is actually very concerning. Why that's the case? So let's see the real data analysis results. So we first evaluate. So we use this spice test and our bootstrap-based methods to evaluate the correspondence between subject-specific cortical thickness and the sucker's depth brain maps. And the spice test always rejects the null hypothesis regardless of the brain pathology used. But we find that using our method, we find that a lot of this correlation actually due to the residuals. So the residuals have a very high correlations, almost 0.4. So this is pretty rare in neuroimaging data analysis. But the correlation between other subject-specific features actually very small. And then we are concerning what's the problem here? So why these residuals are highly correlated? So we first we actually use the same procedures to evaluate the correspondence between subject-specific motor task and language task brain maps. So for these two types of the functional brain maps, we actually find different results. So even though spice tests still get the significant results, but using our method, we actually find that the correspondence are due to different reasons. So for the motor task and the language task brain maps, and we find that even though the correlation between residuals are still significantly correlated, but the magnitude is very small, smaller than 0.1, very close to 0. And a lot of this correlation actually coming from the interaction subject effects, that is the subject-specific spatial variabilities across different locations. So then we think, okay, what's the reason for this different correlation with residuals? So after careful analysis, we realize this is because the cortical thickness brain maps and the sucker depth brain maps are generated from exactly the same structure magnetic resonance imaging. So it's highly likely that these high correlated residuals are due to the same imaging and under analysis. So in contrast, those motor task-based brain maps and the language task-based brain maps are generated from separate fMRI scanning. So that's why there's no correlations that's due to imaging procedures. And we think that it's highly likely that this high correlation residual are mainly due to the imaging procedures, not due to the biological reasons. Overall, you can clearly see that this based on our models, we can clarify different reasons or different variability for brain map correspondence. Okay, let me summarize all the analysis results. So under this uniform statistical model, we show that this actually existing permutation procedures actually have different null hypotheses. And the spin test and the brain smash also rely on very strong and mathematical assumptions. And just based on the model, we can easily have this bootstrap-based inference procedure that achieves much higher power than the existing method. We can also review different forms or different reasons for the brain map correspondence under our models. And this model can be extended to incorporate like subject covariates, age and sex to bring a deeper understanding for the different reasons for the brain map correspondence. And because of time, so let me thank my wonderful PhD students, Chao Chu and Lin Yi. So thank their great work in this project. And Lin Yi actually prepared the slides for me. And I also want to thank my statistical collaborators. So Tom actually first mentioned to me that there's a lack of statistical models for brain maps. So that's why actually there are a lot of confusing about the different method for evaluating brain map correspondence. Dana is a biostatistician at the University of Pittsburgh, and she's a long-term collaborator of mine. And I also want to thank my wonderful MAMO AD research team, and thank Diego, L'Oreal, Emily, Lauren, Alfonso, and Stacy from University of Pittsburgh, and Greg from Jackson Laboratory. Thank their great support and enormous support, and gave me a lot of inspirations about in our studies of normal and abnormal brains. And most importantly, thank everyone for attending my talks and any questions.
Thank you, Tingting. Really great talk. I think right now that's open to questions, and then please raise your hand if you have any question for Tingting. Yes, Anderson.
Hello, thank you. Oops, my camera is open. Okay. Thank you for this fantastic talk. Very interesting, very, you covered all the important topics. You are spot on all the main issues. There is one other method that was proposed recently. It's called eigenstrapping, which can do, in which the brain maps are decomposed into spherical harmonics, and the harmonics are themselves shuffled, and that offers an alternative. But then that method is also invalid for different reasons, because I'm saying this on my own, right? It's not in literature. I'm telling you from my own knowledge. The results of that is going to be invalid as well, because when we shuffle the harmonics and we combine them by the central limit theorem, eventually the data will become Gaussian. But then that doesn't respect the original data, right? But anyway, I'm just pointing out that there is this fourth method. Another thing, it's not really a question, but I'd like to comment for everybody who is in the audience. The spin test is invalid. Never use the spin test. Advise your students, your postdocs, if you're doing any work that involves the spin test, do not use it. And the issues with it are not just what Dr. Tingting presented, that have to do with non-stationarity, but the spin test, not just it depends, it relies on non-stationarity, on the data being stationary, but it's so bad that it actually introduces non-stationarity in the data. Even if you simulate, actually, Dr. Tingting, on this I have a question for you, because your simulations, you simulated perfectly stationary data and you observed good results, but you probably started at the end, your simulation probably started at the very end of the pipeline for the spin test. But when you start with the original data, that inflation process that creates that sphere, that introduces stationarity. So even if the data starts perfectly, like ideal, according to the theory, right, perfectly stationary, when we inflate, some parts of the brain shrink, some other parts of the brain expand, and that happens in different ways. And now we have non-stationarity being introduced there. So the method cannot possibly be valid in any circumstance. So we should never use. Everybody who use the spin test to draw major conclusions in their papers, they should absolutely reanalyze their data, because this method has no hope. Let's say it should not be used. And on top of that, there are two other issues that are specific to it that the other two methods don't have. One is the treatment of the medial wall. So the brain, the cortex on each hemisphere is not a topological sphere, right? It doesn't have genus 0. So, but the spin test ignores that. It just keeps the medial wall there, and that gets shuffled and mixed up with the real data, with the true data, right?
Yes, exactly.
And let me comment one more thing. I know we are short in time, but it sounds like I'm talking more than you, right?
Oh, yeah, I like your comments. Exactly yes.
This is important, yes, because I want to bring this up so everybody knows this, but if it wasn't considered before, maybe it needs to be considered in additional simulations. The third issue is the spin test. So one is non-stationarity, the other is the medial wall. The third one is that of interpolation. So when we inflate into a sphere and we rotate, well, first it's a sphere. You have all the vertices in their places in space, but the brain is sampled discretely. It's not continuous, right? So it's a discrete mesh. So when we rotate, the vertices are never going to align. It's impossible. It's mathematically impossible. The only way in which it could be possible is if the brain were a perfect icosahedral, a platonic solid, but there are only 5 in nature, and that's not clearly the case of the sphere with thousands of vertices as we have. So we have to interpolate. But then here it gets pesky, because the first permutation, in which we don't permute the data, the original data doesn't need any interpolation. But all the other permutations, we have to interpolate. And by
Oh,
interpolating, now the data are no longer exchangeable, right? You violated one of the cardinal rules of a permutation test. So it's really invalid. It shouldn't be used. And the last thing I would like to comment, Dr. Tinting, if you permit, is when you say that you believe that functional and structural, they are capturing different aspects of the equipment, right, of the fMRI and being structured, because there are different methods coming from separate MRI acquisitions. What I believe is truly happening is that some of those modalities, they capture anatomy, brain structure, the actual underlying anatomy that is unique for every participant. And in the case of SPICE, when you have imagings that are dominated by anatomy, when you compute the correlation within subject, so two spatial lines, let's say focal depth and thickness, with the same person, the anatomy matches perfectly because after all, it's the same brain, it's the same person. But when we try to map with, when you permute and do the correlation with a different participant, then of course it's a different brain. Then the alignment is never going to be perfect. And that is what introduces false positives, and it's less severe for functional data, because functional data, even though it comes from the same scanner and the same imaging session, is just a different tuning of the scanner that we use. It's actually capturing something else that does not depend as much on anatomy, which is the bold response or some other aspect of the brain, but anatomy doesn't take a major role there. So I think these are the issues.
Oh,
All right.
yes, that's great. Yeah, that's great.
So
So
yeah.
it's important to have a model, right? If we can separate those different reasons for that
Yeah,
give rise to Honestly, I really like your comments. Exactly. Yeah. It's just that I find that spin tests have been used so extensively.
yeah, it's crazy, right? And that's because so it's very difficult to create, as you know, here in this audience, most of you are statisticians, so you know how difficult it is to create new methods. And we know that the spin test is invalid. We have known this since at least 2019 in my group, and we have been discussing this, and we know, but it's very difficult to publish a paper criticizing without proposing anything better. And what I really like about your talk and your work, Dr. Tinting, is that you are proposing something new. And this thing that you are proposing has many qualities, right? Has many advantages compared to what everything else has been doing. But because it's hard to publish just criticizing a method and not telling anything better that can supersede that, then nobody publishes the result just criticizing others. And then the field as a whole never ends up finding out that the method is invalid, that they should not be using. And then you have all these many, hundreds of papers with, this done so
Exactly.
badly, right? It's really bad. It's a disaster, actually. Well, anyway, I like very much your presentation. And one question then actually I do have. You mentioned that with the model that you're proposing, we can add additional covariates, right? Let's say age and sex and all this. Couldn't this be confounded perfectly with the subject-specific term? You have, in one of the earlier slides, you have a term that is for the subject-specific effects. If we put, let's say, age, sex, and those things, isn't there a risk that those subject-specific terms could be perfectly confounded with those subject-specific terms that are already in the model? I'm not sure. So this is a question.
Exactly. You are. Anderson, first, I want to say I really like your comments. For me, it's more from the mathematical understanding, and your understanding of imaging is excellent. So exactly. So we think that if we can, so because we categorize subject-specific feature in beta and omega, so it's very likely that if we can associate the beta with subject covariates like age and sex, we can if we find the beta have a largely can be explained by subject covariates like age, sex, or disease status. So this also indicates that those features in brain maps actually exactly reflected actually some confounding factors that drive the correspondence between the brain maps. Yes, exactly. Yeah.
All right. Thank you. Thank you.
That's excellent comments. Thank you so much.
Yeah, thank you.
Great. Thank you for the discussion. I think maybe due to time, so then if you have any additional questions, maybe directly email Tingting. And then Tingting, can I share my screen again?
Yeah. Let me stop share. Yes.
So thank you, Tingting, for the great talk, and then also the great discussion, and then just to give you a preview for our next seminars in April, we will have two seminars. One is from Dr. Mingyao Li, and then we will switch the gear a little bit. She will talk about her research on single cell and spatial genomics, and then we will have another webinar in April from Dr. Josh Siegel, because last time when Dr. Robert Cass talked about Allen Institute and many show interest to learn about research there. So that's why we invited Josh to give a presentation to the group as well. So next month will be a busy month, but I hope you can join us again next time. So, and then thank you, Tingting, again, and thank you all for joining us, and then hope to see you next month. Bye.
Yeah, thank you, Aga. Yeah, and Pan.